What I'm going to talk to you about today are T cells, B cells, and viral factors that are involved in generating neutralizing antibodies. So I'm going to talk about two things. First of all, why neutralizing antibodies? Why we think they're important. You've heard a lot about T cells. You've heard about non-neutralizing antibodies, things that can interact with mucus, etc. And then I'll go on to describe a system where we can actually start to break apart what's important for the generation of broadly neutralizing antibodies during natural infection. So, why neutralizing antibodies? Well, if you go back to the vaccine, the history of HIV vaccines, there was this vaccine developed by Merck. It was an Ad5-based vaccine, and it expressed GAG, POL, and NEF. So it didn't express any envelope. It didn't generate any neutralizing antibodies. It was based upon the concept that you needed to stimulate CD8 T cells and that would then protect the individual from infection. And here were the results of that trial. If the participants had low pre-existing titers to Ad5, there was no protection. In fact, if they had high pre-existing neutralizing antibodies to Ad5, the vaccinees actually did worse than the placebo recipients, and I won't go into that. But basically, it failed miserably. Now, the concept of this vaccine was not actually to protect against infection, but to control viral load once people became infected, but there was also no effect on viral load. So this really, I mean, it's not the death knell for CD8 T cells. You heard from Ashley Haas about some exciting data about CD8 T cells and protection. But there have been two subsequent vaccine trials. This one, which was an ALVAC that expressed GAG, POL, and envelope that was boosted with envelope protein. And it was published in 2009, and what you can see is the placebo recipients became infected. The vaccine recipients became infected less frequently. This was about a 31% efficacy. So here, where you actually developed antibodies against HIV envelope, there was some protection. A subsequent trial that was a DNA prime Ad5 boost, which expressed envelope, GAG, POL, and NEF. So it was designed to stimulate both CD8 T cells and envelope antibodies, showed no efficacy. These placebo and vaccine arms really overlap. So here we had protection, here we didn't. Both of these generated antibodies. So what was the difference between these? Well, a very extensive correlates analysis was done on this trial, and that was published just about 2 years ago. And the bottom line of that analysis was that they looked at 6 different things: IgA binding to envelope, avidity of binding to GP120, ADCC activity, neutralizing antibodies, binding to V1, V2, and CD4 T cells. And 2 things were statistically significant, but I think what's probably the most important is that immunoglobulin that bound to the V1, V2 region of envelope was protective, about 43% protective in this correlates analysis. Now, here are the actual data. If you were a vaccinee and you had either low or medium titers to the V1, V2 region, here's your risk of getting infected. If you had high titers, you had a much lower risk of getting HIV infected. These data have been looked at over and over again. There have been multiple follow-up studies from this initial analysis, all of which are listed here, all looking at V2, V1, using different assays, using circularized peptides, using overlapping peptides, and all of them confirm this initial data, that antibodies to the V1, V2 region, mostly the V2 region of the envelope, really were associated with protection. Now, this doesn't mean that's the mechanism, but certainly it's very intriguing that antibodies to this region of envelope seem to be associated with decreased risk of infection. The reason I say this is interesting is we have now been able to identify regions of the HIV envelope that are targets of broadly neutralizing antibodies, and there are 4. One is the membrane proximal external region of GP41, shown here. One is the CD4 binding site. One is a glycan-dependent V3 site on GP120. But the one I'd like to draw your attention to is this. The V1-V2 site on GP120 is the target of broadly neutralizing antibodies. And there have been multiple broadly neutralizing antibodies that target this site. So the correlate was in this vaccine trial was the same region where we know broadly neutralizing antibodies can target. Now, one thing about these broadly neutralizing antibodies that I'll come back to is that most of these exhibit a very high rate of somatic hypermutation. So they have to go through a lot of maturation during their development. So now if we go back to those 2 clinical trials, the ALVAC or the TIE trial, RV144, had protection. HVTN 505 did not have protection. Well, why was that? So these are the antibody titers in HVTN 505 where there was not protection. And what you have here are 4 different GP140s. Now what you see is that all of the vaccinees pretty much developed very good, very high titers against GP140. But if you look at the V2 region, what you see is only 20% against this V2 protein, or 17 against this V1, V2 protein, developed any antibodies, and it was at much lower titers than against the GP140. And this just shows the data against here 3 different V1, V2 proteins, comparing the RV144 trial, where there was protection, and the HVTN 505 trial, where there was not protection. And what you see is many fewer individuals in the 505 trial generated an antibody response to this region, and when they did, it was at much lower titer. So that's probably why the HVTN 505 trial failed. We didn't induce antibodies to this critical region. Now, another bit of evidence that neutralizing antibodies are important is the fact that you can actually do proof of concept studies. So you can take broadly neutralizing antibodies, you can infuse them into monkeys, and then you can challenge those monkeys. So in this case, the only immune response against HIV that these monkeys have is the antibody that you've infused. And there have been several papers in the past published on this. Here's a broadly neutralizing antibody B12, here 2G12. When you challenge these monkeys, you infuse these antibodies, you challenge these monkeys with SHIV, so these are SIVs, but they have an HIV envelope. These monkeys are protected. Now, these 2 antibodies which were used are really the older broadly neutralizing antibodies. They're shown here. Subsequent to that, we've isolated very broad neutralizing antibodies from multiple patients. And what is shown here is what we call a potency breadth curve against 190 different viruses. So you test each antibody at multiple concentrations against 190 viruses. And for each virus, you come up with what's the IC50, what's the concentration at which you neutralize that virus 50%. And then you just total that up. And what you see is that we now have these antibodies that are much more potent and much more broad than these 2 that we know actually protect monkeys from infection. And so let me just show you some data with VRC01, which is this one. It's sort of right in the middle of all of these broad, potent neutralizing antibodies. This is a CD4 binding site antibody. And here, whether you do a rectal challenge or a vaginal challenge after you infuse this antibody, what you see is that in the absence of the antibody, none of them are protected here. Only 1 of 4 was failed to get infected after the vaginal challenge. But all of the monkeys were protected when they got the BRCA1 infused as compared to a control. So, broadly neutralizing antibodies in and of themselves, all by themselves, can protect against both rectal and vaginal transmission. And finally, the last bit of data I'd like to give you on this is that many individuals develop broadly neutralizing antibodies. Somewhere on the order of 15 to 25%, depending upon how you define the breadth of neutralization, will over time develop broadly neutralizing antibodies. And what you can see is it really doesn't matter whether you're a long-term non-progressor, a slow progressor, or a progressor. You know, these antibodies tend to come up in about up to a quarter of individuals. But these broad neutralizing antibodies don't actually provide any protection or any benefit to the individuals who are infected and who generate these antibodies. So what's really important is to have these types of antibodies there before you're exposed to virus, because then we know they can protect. But just like with antiretroviral therapy, if you generate these while you're infected, your virus just tends to escape from them. So from this part of the talk, you know, a vaccine designed to stimulate only T cells and not functional anti-envelope antibodies failed to protect. Among two vaccines that stimulated on-specific antibodies, only the one that generated antibodies against V1, V2 was protective, and these antibodies correlated with vaccine protection in multiple follow-up studies. We know that this region is a target for broadly neutralizing antibody, and passive transfer of broadly neutralizing antibodies protect monkeys from Shiv challenge, even though they offer no benefit to subjects from whom they were isolated. So let me move on now and talk about what are the factors associated with the development of broadly neutralizing antibodies. I think for a vaccine, while I agree that maybe non-neutralizing antibodies may be able to protect, and I'm sure you'll be hearing data about that in coming years. You know, if it were me, I would like to have a vaccine that stimulates neutralizing antibodies. So, what does it take to generate a neutralizing antibody? Well, I think the nice thing to do is to study some of these individuals who generate broadly neutralizing antibodies and dissect what's associated with the generation of broad neutralizing antibodies. That's sort of a long, difficult, and arduous process. But what we can do actually is go to a animal model. So several years ago, in the development of Shiv viruses, Mal Martin and his group identified a Shiv called Shiv88. It's based upon a CCR5 using HIV envelope derived from ADA. He noticed that among these animals, after they got infected and were followed for a long period of time, he had 14 monkeys, and one of them developed very broad neutralizing antibodies. So now we have a system where we can infect monkeys, we can follow them, and see what's associated with these monkeys that develop broad neutralizing antibodies. So in doing this, one thing that we want to look at is what's going on in the lymph node and what's the interaction between T follicular helper cells and B cells, because we know that where antibodies are developing, maturing, is in the B cell follicle area, and specifically within the germinal center. And within this region, we have specialized T cells called T follicular helper cells, which upregulate CXCR5, downregulate CCR7, and move into the B cell regions of the germinal center, where they interact with germinal center B cells. And through this interaction, they liberate IL-21. They interact with B cells through a number of different molecules. And this induces somatic hypermutation, usually through the upregulation of AID, activation-induced deaminase, in the B cell. So this is something that's going to be critical. This interaction is going to be critical when we study these monkeys. Another reason that this is going to be, we think this is important, is that my group and Hendrik Streek's group published just last year in SIV infection and HIV infection that these T follicular helper cells in SIV-infected monkeys and HIV-infected humans are often increased. So the generation of these T follicular helper cells, where you've heard about Th17 cells and all sorts of other CD4 T cells being depleted by HIV and SIV, in many cases, these T follicular helper cells are actually increased. So they're not depleted, but actually accumulate during lentiviral infection. So here's the experiment. We take 8 monkeys. We do a lymph node biopsy. We then infect them intrarectally with Shiv-88, and then we do multiple lymph node biopsies thereafter and also collect blood and plasma on these monkeys. We know from past studies that it's going to take 40 to 50 weeks before we get neutralizing antibodies. So at 44 weeks, we look at the serum of these monkeys. And that's, here are the 8 monkeys over here. And we test them against a panel of about 20-some different viruses. And we ask how well is the sera from these different monkeys neutralizing these different viruses. And we come up with a neutralization score which measures both breadth and potency. And what we do is we say, okay, if you have a titer against a specific virus of 40 to 100, we give that a 1. Those are the ones shown in green. If it's 100 to 1,000, we give it a score of 2. If it's greater than 1,000, we give it a 3, and we just total that up. And what you see is that of these monkeys, after 44 weeks, 3 of them had scores greater than 20. So those we call our good neutralizers. 3 had scores between 10 and 20. Those are our intermediate neutralizers, and our weak neutralizers had scores less than 10. So in these 8 monkeys, we had 2 weak neutralizers, 3 intermediate, and 3 strong or good neutralizers, and they'll be shown throughout the talk as red for good neutralizers, blue for intermediate, and black for weak. First thing is, how is this associated with viral load and CD4 count? So in the blood, what you see is the good neutralizers had the highest viral load. The weak neutralizers had the lowest viral load. And the opposite for CD4s. The ones who had the best neutralizing antibodies were the ones who had depleted CD4 the most, and those who maintained their CD4s had the worst. So you really need a lot of antigen stimulation to generate broad neutralizing antibodies. Then we decided to look in the lymph nodes at T follicular helper cells. So here we just look at the central memory cells, those that express CXCR5 and PD1, those are your T follicular helper cells. And we map this out, and we can see that maybe the broad neutralizers and the intermediate neutralizers had a little better accumulation of T follicular helper cells, but it wasn't as clear as these viral load results. So this wasn't the entire story. So then we decided that what we really needed to do was look at antigen-specific T follicular helper cells and B cells in the lymph node. So once again, we can gate on the T follicular helper cells. We can then stimulate those cells with envelope or no stimulation. We can look at whether they mobilize CD154. And within those cells that mobilize CD154, are they making T follicular helper cell-like cytokines, IL-4, or Th1-like cytokines, interferon gamma? Usually for T follicular helper cells, you'd want to look at IL-21. The staining doesn't work very well in the lymph node, so I don't show those data, but we'll use IL-4 as our marker of T follicular helper cell function. In addition, as Adrian McDermott had told you the other day, you can gate on B cells, in this case within the germinal center, and you can see if they bind probes, GP140 probes, and what you can see is over time you get more and more of these B cells in the lymph node. So now if we go back and say what correlates with the generation of good neutralizing antibody, here we're looking at B cells. What you can see is envelope-specific B cells in the peripheral blood are a poor predictor of neutralizing antibody. But in the lymph node, they're extremely good. So if you've got envelope-specific B cells in your lymph node, the more that you have, the better your neutralizing score is going to be. What about T follicular helper cells? So if we just look at envelope-specific T follicular helper cells that respond by mobilizing CD154, there's a reasonable correlation. But what happens when you break that down by those that are making IL-4 versus interferon gamma, you see that the correlation is really good with IL-4, not good with interferon gamma. So what this tells us is that you have cells within the lymph node that are CD4, they respond to envelope, they are probably in the germinal center because they have the right phenotype to be in the germinal center, but they may not be making the right cytokine. So they may not have the right quality. So to better look at the quality of these cells that we're calling T follicular helper cells, we sorted 200 envelope-specific CD154 positive cells from each one of these monkeys in duplicate, isolated the RNA, and performed quantitative RT-PCR, looking at 11 different cytokines and markers of T follicular helper cells. And when we did that, and then just created a tree diagram, what you can see is that the good neutralizers all had upregulation as compared to the other, either the weak or the intermediate neutralizers, of the really good T follicular helper cell markers, such as BCL6, the marker of T follicular helper cells, CXCL13, MAF, IL-21, and they actually had lower expression of like TH1 type of markers, TBET, BLIMP1, interferon gamma, FOXP3. So within the T follicular helper cell population, there's a gradation in quality of function. And the better the T follicular helper cell function, the better your neutralizing antibody response is going to be. So I'd like to go back and show you this one more time. This is the envelope-specific IL-4 producing T follicular helper cells versus neutralization score. This is a pretty good correlation. The reason I bring this up is over the last couple of years there have been multiple papers on, instead of looking for these T follicular helper cells in the lymph node, looking for them in the peripheral blood. So here's one, here's another, here's another, here's another. Clearly, being able to measure these types of responses in the blood is a lot easier than pulling out lymph nodes. Well, if we now take these publications, look at the markers they use to define peripheral T follicular helper cells, we can say, how well did that do, how well did those things predict neutralization in the monkeys? Well, the problem with these papers is that they all use different panels. So here are the 4 papers I just showed you. They're all CD4 positive. In general, they're CXCR5 positive except for this paper. In this case, 2 of them are CXCR3 negative. This one's CXCR3 positive. Usually they're PD1 positive. ICOS can either be positive or negative. But what we decided to do is to say these 2 papers tend to have the same panel. This has a different panel. Let's apply these markers to our monkeys and see how well they predict neutralization. So here's the gating, the panel we put together to identify the CXCR3 minus PD1 positive, CXCR5 positive central memory cells, and here's the correlation with neutralization score. It's pretty bad. If we then instead went for the CXCR3 positive cells and looked at those that were PD1 positive and ICOS positive, once again, the correlations are not good. So to answer the question, how well do these peripheral T follicular helper cell definitions perform as compared to looking within the lymph node, basically they don't perform. So if you want to look at T follicular helper cells, my feeling is you have to get lymphoid tissue because that's where the actual interaction is going on. So what other factors can we look at that are associated with broadly neutralizing antibodies? Well, we know that there are some common features of broadly neutralizing antibodies: extensive somatic hypermutation and long CDRH3 regions. So let's first look at extensive somatic hypermutation. So what we did is we sorted individual memory envelope-specific B cells from week 44. This was done from the bone marrow on all these different monkeys. We sequenced the rhesus immunoglobulin gene, heavy and light, from individual cells, so we would be able to hopefully go back and pair the heavy and light chain sequences. And we compared these gene sequences to a rhesus immunoglobulin sequence database that has recently been developed by Tom Kepler after Danny Dueck's group resequenced this locus in rhesus monkeys. So when we did this in our 8 monkeys, here's what we see. So each one of these is a B cell heavy chain gene. So this is an envelope-specific IgG gene from an individual B cell. And here you can see the numbers. There are 150 from this monkey, 61 from this, 108 from that. And what you can see is, in general, I mean, there's a big spread in terms of somatic hypermutation. But if you look at the mean level of somatic hypermutation, what you can tend to see is that the better neutralizers have a higher mean percent of V heavy chain gene somatic hypermutation than the weak neutralizers. And here's the correlation. Quite good. And now what we can do is look at the one monkey that had the best neutralization score and the highest level of somatic hypermutation, and actually start looking at some of these individual sequences. But before we do that, we have to identify, we wanted to identify the potential epitopes that were targeted. So Evelyn Georgiev published last year in Science that if you take sera and you test it against a big panel of viruses that were specifically chosen based upon sensitivity of these different viruses to the different classes of neutralizing antibodies. So you take some viruses that we know are sensitive to CD4 binding site antibodies. You take other viruses that are sensitive to MPER antibodies, etc. What you can do is look at that sera and you can deconvolute it, and you can say this sera is probably targeting this epitope or that epitope. So we did that with our best neutralizing monkey, and you really need really good and broad neutralization before this works. But what we can see is that this monkey over time appeared to generate a PGT128-like response. So this is a V3 glycan targeted response. Now we know that those antibodies tend to have reasonably long CDRH3s. So what we could do is in this monkey we could go back and say what were the CDRH3 lengths of the antibodies in this monkey. And what we found is that, on average they were around 15, which is standard. But there were there were several B cells that had 30, 31 amino acid CDRH3s. These are very long CDRH3s. And interestingly, all of those that were 31 amino acids in length were clonally related. They were all from the same V heavy chain and all used the same V light chain. And these that were 30 amino acids in length were also clonally related. I mean, there were obviously somatic hypermutation, somatic mutations within that, but it was all the same heavy chain usage. Now, the total somatic hypermutation in these two clonal families of antibodies wasn't all that high. It's around 15% somatic hypermutation. And these weren't the most somatically hypermutated. But the nice thing is that we could pull out a heavy chain and a light chain, and we could actually make an antibody and ask, does this antibody neutralize? And if so, what's it targeting? So we haven't completed all of this. We did make this antibody that we pulled out of this monkey. And what you can see is it does neutralize quite well ADA. It neutralizes quite well this other virus. It doesn't hit JRFL that the serum hits. So it doesn't completely recapitulate the serum neutralization, but it is clearly a neutralizing antibody, and it's got at least some of the breadth that we're seeing in the serum. The other thing that we wanted to do is to look at the contribution of the envelope to the generation of broad neutralizing antibodies. And basically there have been human studies that have been done, in this case by Bart Haynes, where they've looked at envelope sequence variation and the generation of broad neutralizing antibodies. This was a patient who was identified soon after seroconversion and who developed fairly broad neutralizing antibodies to cover about 55% of viruses to the CD4 binding site. And this occurred over 2 years, and they studied the evolution of both the envelope and the neutralizing antibody over this period. This shows the antibody tree. So here's the unmutated common ancestor, and what you can see is there are all these different antibodies that came off of it in these 3-box bar charts, I guess you'd call them, are the neutralization ability against 3 different viruses. So if it's red or orange, it's really good neutralization. And what you see is if the antibody mutated in this direction and followed this course, you ended up with an antibody that, or a couple of antibodies that had very good broad neutralization. However, the antibody could go in different directions. It could come off this way and never generate really good neutralizing antibody. So how did this relate to envelope? So they also sequenced the envelope over time. And this is a very complicated slide, but what you have on the top of each, so this is week 14, 22, 30, 53 of infection in this individual. The top is the GP120, the bottom is the antibody. In the GP120, if you have red areas, that's where sequence variation occurred over time. All these colored balls on the antibody are areas of somatic hypermutation. Now, if you study this for several hours and don't fall asleep, what you will notice is that, so there's not a lot of mutation that's occurring over here. And then you see this area on the GP120 that mutates. And the next time point, you have all of this somatic hypermutation within the antibody near that region, and this region starts to go back. And then what you can see is over here, you see a little red showing up here, and you get a lot of somatic hypermutation over here. And what clearly appears to be happening is that there's coevolution and this interplay between the envelope mutating, probably to escape the antibody. The antibody then hypermutates so it can now recognize that new envelope. It drives the envelope back, and there's this constant interplay and this interaction between the antibody and the envelope that is probably going to be necessary to get this degree of somatic hypermutation. In the absence of the antigen, your GP120 evolving, you're going to get to a certain degree of mutation. You're going to have a really avid, high affinity antibody, and it's not going to mutate any further. So we looked at the envelope in these 8 monkeys. So we did envelope sequencing. Well, Brandon Keel did this. And basically, here's what we found. So this is the envelope trees for our 2 really poor neutralizers. Here are the envelope trees over 80 weeks for our two of our three intermediate neutralizers, and as you can probably imagine, these are our good neutralizers. So the envelope was constantly evolving in the three monkeys that generated really good neutralizing antibodies. And this is the monkey that had the best neutralizing antibody. And what you can see is it looks as though out of one corner of the envelope tree, you get a new envelope at week 35. And then out of one corner, you get a new envelope that shows up at week 60. And out of one corner, you get an entirely new envelope that pops up at week 80, as though something's driving the envelope to change this dramatically. So the question is, all of these sequence mutations, are these within that epitope that we had potentially identified, the V3 glycan region? So here's a crystal structure of the V3 glycan. There are two glycans that are key to this interaction, the N301 and N332 sites, and then there's a lot of other interactions in between. So if we look at that region of the envelope in this monkey over time, what you see is the glycosylation sites, N301 and N332, don't change. So they're maintained. But what you also see is in between, starting at week 35 and continuing through week 80, there's tons of mutations right in that region where we know there are these other interactions of the antibody and the envelope. So it really looks as though the envelope is mutating in response to the antibody that's been generated. And there is this same type of interplay that was seen in Bart Haynes's group. And we were actually making some of these envelopes, and they're being tested against the antibodies that we've pulled out of these monkeys, so we can start to actually map whether that's what's going on. So that's as far as we've gotten with this story now, but I think that what we can clearly say is that the SHIV-88 infection, this can be used to study factors associated with the generation of broadly neutralizing antibodies. Broadly neutralizing antibodies develop slowly during chronic infection, and they're associated with high viral loads, low CD4 T cell counts, accumulation of envelope-specific T follicular helper cells and germinal centers in the lymph nodes. Please don't go looking in the peripheral blood. You're going to be disappointed. Greater regions of greater somatic hypermutation within the antibodies, and evolving envelope sequence diversity, and these two are probably quite closely linked. And therefore, what we think is that antigenic changes will be required for immunogens to drive the degree of somatic hypermutation required for broad neutralization. In addition, the fact that you need these very high viral loads probably means that we need chronic antigen expression in our vaccines. And we're not sure whether chronic antigen expression in the absence of the envelope sequence diversity is going to be sufficient. But I think these studies inform the design and protective efficacy testing of the envelope immunogens that are being put into different viral vectors and are being used as protein immunogens, both now and in the future. And so I'd like to just end and thank a number of people. Takuya Yamamoto was involved in all of the SHIV studies, along with Rebecca Lynch from John Mascola's group. I told you about Evelyn Georgiev and Danny's group doing the rhesus immunoglobulin resequencing, and obviously Mal Martin's group for the virus, and a lot of help with the SHIV system. Brandon Keel for envelope sequencing, and our lab animal medicine group. So once again, as I mentioned to you, the best science is done as a collaboration. Every single study that you've seen presented here, there's a long list of collaborators, so keep that in mind as you choose your career path. Thank you.